Uh, I'm Shinya Uchida from Shizuoka, Japan. So I'm an associate professor in University of Shizuoka. So I come from Japan, Shizuoka, Japan. So many people, everyone know Tokyo and Kyoto, but a few people know Shizuoka. So Shizuoka is located between Tokyo and Kyoto. We can see Mount Fuji from our university, like this photograph. So I'm also interested in pharmacy education because in Japan also pharmacy education is changing dynamically, and we have 80 students a year in our college. But so I'm today I'd like to talk about another interest. I'd like to talk about simultaneous evaluation of the activity of 5 cytochrome P450 enzymes by a cocktail study in healthy volunteers. So cytochrome P450 CYP are involved in the metabolism of many drugs that are clinically used worldwide. Approximately 50% of the medicines are metabolized by CYP3A4. However, CYP1A2, 2C9, 2C19, and 2D6 also play important roles in drug metabolism. The activities of CYPs are reported to show large inter-individual variation. In addition, certain medications can induce or inhibit their activities, resulting in drug-drug interaction. Thus, determine and predict potential drug interactions is important for dose optimization. There are multiple approaches for studying drug-drug interaction that focus on CYP activities, including in vitro study, modeling and simulation, in vivo pharmacokinetic study using animals, and clinical study, clinical PK study, pharmacokinetic study in healthy subjects or in patient and special populations. Among these various approaches, evaluation by clinical study has the greatest impact. However, multiple drug administrations are required for determining the activity of each CYP isoform. On the other hand, a cocktail approach can measure the activities of multiple CYPs. In this approach, cocktail of multiple CYP-specific probes are simultaneously administered, and the activities of CYPs are evaluated by measuring pharmacokinetics of each specific probes. Since the Pittsburgh cocktail was first introduced in 1797, several cocktails, including Karolinska cocktail, Cooperstone cocktail, and Inge cocktail, has been developed. However, assay techniques in these cocktail studies do not permit the simultaneous analysis of multiple CYP substrates and their metabolites. Thus, in this study, we first aim to develop and validate a rapid and selective LC-MS-MS method to determine the plasma concentrations of 5 CYP probe drugs, and by a single-step extraction followed by a single LC-MS-MS run. In addition, using our cocktail method, we aim to clarify the chronological changes in rifampicin-induced CYP activity after rifampicin discontinuation. First, we developed a general assay that could simultaneously detect all the CYP probe drugs. This slide shows CYP probe drugs and their metabolites in our cocktail study. Caffeine, losartan, omeprazole, dextromethorphan, and midazolam were used as a substrate for CYP1A2, 2C9, 2C19, 2D6, and 3A4, respectively. As shown in this flowchart, in sample preparation, we used an Austro 96 well plate for the extraction of series of CYP substrate and metabolites from human plasma and urine. In the case of urine, the samples were added to the plate after the treatment with beta-glucuronidase. Following optimization of the chromatographic condition, all the peaks were separated, and retention times range between 4.4 and 11.7 minutes, and total run time for a single injection was within 13 minutes. The precision and accuracy values suggested that the assay has high accuracy and reliability in plasma and urine samples. No significant matrix interference was observed. To demonstrate the clinical application of the assay, plasma and urine concentrations of 5 CYP probe substrates and their metabolites were determined after simultaneous oral administration of 5 drugs to 4 healthy volunteers. All the substrate and metabolites were determined over 8-hour period, and the plasma concentration of each substrate at each hour after administration were above the lower limit of quantification. So we have developed and validated a rapid and selective LC-MS-MS method. Then we aim to clarify the chronological changes in rifampicin-induced CYP activity after rifampicin discontinuation using our cocktail method. 13 healthy volunteers took rifampicin 450 milligrams once a daily, and the cocktail drug was repeatedly administered before the rifampicin treatment, and 24 hours, 4 and 8 days after the rifampicin discontinuation. This slide shows the time courses of plasma concentrations of caffeine, losartan, omeprazole, dextromethorphan, and midazolam after the administration of cocktail drug. After the administration, the plasma concentration of 5-CYP substrate and their metabolites were determined until 8 hours, and pharmacokinetics parameters of drugs and metabolites were calculated from these plasma concentrations. As shown in this slide, the plasma concentration ratio of omeprazole to 5-hydroxyomeprazole was significantly reduced after rifampicin treatment and returned the baseline level 8 days later. Similarly, the AUC ratio of midazolam to 1-hydroxymidazolam was reduced on day 0 and day 3, and on day 7, the reduction was returned. These results suggest that activities of CYP2C19 and CYP2C3A4 were induced by rifampicin treatment, and the inductions remained at 4 days and returned 8 days later. So in conclusion, we developed and validated a rapid and selective LC-MS/MS method to determine the plasma concentrations of 5-CYP probe drugs and metabolites by single-step extraction followed by a single LC-MS/MS run. This assay had high accuracy and reliability for plasma and urine samples. And the induction of CYP2C19 and CYP3A4 activities by rifampicin remained at 4 days after the rifampicin discontinuation and returned to baseline levels 8 days later. So our cocktail approach enables in vivo assessment of the activity of various drug metabolizing enzymes and the detection of potential drug interactions in a single assay. These are the people with whom we have collaborated. Especially, a clinical study was done in collaboration with Professor Watanabe and Dr. Inui at Hamamatsu University School of Medicine. And Shimako Tanaka mainly performed the development of LC-MS/MS method. Thank you for your attention.
So, are there any questions? I unfortunately got your presentation halfway, but I'm interested in the method that you use to determine those metabolites, because I work with coffee and I look into caffeine and I look to genotyping and the CYP1A2. So, and I'm exactly right now going to this gold standard method to measure metabolites. So, do you have your method is already published to do that you validated and all that? So, the LC-MS method?
That report was published already, yes.
And is that one of those that you put on the bottom of the screen when you were showing the data? Was published in that magazine that was showing the bottom of the screen?
Yeah.
International or something. Can I get that from you, the reference? I'm interested in the reference. Yeah.
Okay, so assay method was published in this.
Okay, 2014, all right. Mm-hmm. Thank you so much.
Thank you.
Thank you. Are there any other questions? No.
Thank you very much. Thank you.
Thank you very much.